没点，我说这个前言不能一样啊。Oh,这个写的这个一样，把它说的。Yeah, I heard about it. 所以这是跟那个思路很像的。He's just smiling with that. Yeah. 那个没说到最近这个事情，那我那个 exactly. Okay. It's fascinating. Many of that kind of discussions. 就是这些指标因子。Managing 这个做不了检验，所以只能做一个。所以它就是一个定性。Yeah, cool. 然后这些关系这些关系是错的还是反的，没有改变。No, I guess it's like crazy.
Yeah, I was confused with that.
还有一个加在这儿的定义是 no.
We're trying to, you know.
但是确实到后面根据这个结果，这边没有结果上次末尾的说大家都这样的，是吧？对，他们这个就是他们讲的，所以现在就是结论不太明确。Porter 的建议是 ，Porter 里面提出来，给他提炼出来两个这个有影响的结果，所以应该是关于这些东西。然后我开始给他改前言，更强调这个，但后面到了结果问题了，就变成各大家各种就比较乱了。
But then that's sort of how Ben also started being really interested in the beginning. It was like right when we got back.
What is fixed time?
Yeah, I'm so uncomfortable anyway.
Yeah, I mean, that's my, I've never been here.
Yeah, right.
Some period of time. Because of that, sign from something else for a time and functions and your delays carefully. Yeah. It's really nice.
Okay, good morning, everyone. Thank you all for coming along on the last morning of the workshop, although I should once again specify that this is not technically part of the workshop. This is a series of our longer running series of talks by our thematic sciences, the theoretical sciences visiting scholars. Yeah, it's nice to see all of you here after last night's festivities. I'm particularly glad to hear that nobody was married last night at the bar, so that's nice. So anyway, it's my absolute pleasure to hand over to Rachael Winfree, who's a professor in the Department of Ecology, Evolution, and Natural Resources at Rutgers University in the US. Rachel's been doing response diversity work for quite a few years. That's how I first came across Rachel's name. And I think maybe all of the papers I've ever read about watermelons are published by Rachael. So I'm looking forward to hear about more of your work, Rachel. Please take it away.
Oh, yeah. Should just be connected to Zoom. Yep.
Perfect. Okay, thank you. Thank you so much for that introduction. Yeah, I never set out in my career to work on watermelon. It just kind of happened. But, so thank you so much for this entire workshop. I just want to say to everybody, I've really enjoyed this week, and I've learned so much from all of you. So thank you so much. So since this talk is sort of outside the workshop, I thought what I would do, it's about my research program in general, but what I did was just pulled out things in my research program that are basically about the relationship between biodiversity and ecosystem functioning or services more generally, since that is the larger context for everything we've been talking about with regard to response diversity. So that will be what I'm talking about. So I first want to just say a few words about sort of what my lab does and our approach to our research. So my research program investigates the causes and consequences of biodiversity change. And all of us in my group are ecologists, but we work across a couple subfields, mainly biodiversity, species interactions, and macroecology. And a key feature of our work is that I study biodiversity in nature. These are just photos of me and some members of my research group collecting our data. And we work in both human disturbed and natural systems because both are relevant to the field of ecology. And a second feature is that I believe in data. I am primarily an empiricist, and I actually thought as one of the token empiricists here, I would focus this talk on sort of data collection and what our data actually tell us about some of the theories about biodiversity and ecosystem functioning. And by the way, I'm going to use ecosystem function and ecosystem services synonymously here. I know they're slightly different, but the distinction doesn't matter for what I do. And lastly, I study insects, which from a biodiversity point of view are really the neglected majority. So Bob May pointed out decades ago that to a first approximation, all species on Earth are insects. That is true, but they're vastly less studied than vertebrates or plants. For example, about 80% of insect species are estimated to not even have been discovered and named yet. So the biodiversity loss crisis, most people aren't aware of this, but it actually is an insect biodiversity loss crisis. All right, so today's talk is going to be sort of an overview of what we've learned from our data about biodiversity and ecosystem services in the real world. And by real world, I just mean free living plants and animals that are studied at large scales of space and time. So this question of how biodiversity contributes to ecosystem services has really been one of the larger, more active fields of ecology for the last few decades. But most of what we know about this comes from smallish scale experiments, and the theory is also largely based on these experiments. And many hundreds of such experiments show that in general, as you increase the number of species, in other words, species richness, which is the most commonly used metric, your ecosystem function increases, but it tends to asymptote at smallish numbers. And a few of the key features of these experiments, so they vary species richness, as you all probably know, so just the number of species that go into each of these plots is controlled by the experimenter, but they randomize species composition, so the identity of the species that goes in is generally randomized. And in terms of the mechanisms behind this overall asymptoting curve result, there are two that are about equally important, and I'm obviously generalizing a lot right now, but they're basically niche complementarity, so that the species in the plot contribute to the function differently, or they use different resources to get more function when you have more species. And then roughly equally important is more of a probabilistic thing, the sampling or identity effect, which posits that there are some species are just better at the function than others, and your probability of having one of those species in the plot increases when you are putting more species into the plot. So I'll come back to these ideas because part of my research is about determining whether these mechanisms operate in nature. They operate something different. So my work again is about biodiversity and ecosystem services relationships in what I call real world ecosystems or real world landscapes. So much larger scale. So by large spatial scale, I mean tens to thousands of square kilometers. So really many orders of magnitude larger. And again, naturally assembled communities. So these are unmanipulated communities, whether they're in human disturbed areas or not. And surprisingly, the answer to this question of how species richness affects ecosystem function really is not a consensus answer to that for the real world. So I'm going to argue that there's three features of real world systems that are really important to determining what this relationship is, and that those features have mostly been excluded from experiments in theory. So the first is just aggregate abundance. In other words, the total number of individuals you have in the community, regardless of what species they are. And this has been excluded, I think, indirectly in many of the experimental work, simply because area is controlled, for example, which sort of establishes an upper limit, or resource supply rate is controlled. Whereas in the real world, aggregate abundance varies pretty dramatically across space and time. Another reason aggregate abundance has been excluded is simply the metrics that we use as ecologists to measure biodiversity. So species richness doesn't include aggregate abundance, and neither do indices like Simpson or Shannon, because they're both basically dividing out aggregate abundance, right? They're based on relative abundance. Then the second feature is dominance and rarity. So in other words, a species abundance distribution in real world communities. And these are always highly skewed, with a few common species and many rare ones. But that is not always the case in experiments or theory. And then third, this is just an outcome of working at large spatial and temporal scale. So we're not just looking within communities, we're looking across ecological communities. So we need to think about species turnover in space and time, in other words, beta diversity. So why does any of this matter? It matters because it's often simply assumed that the relationship between biodiversity and ecosystem services is very strong in the real world. So for example, the IPBES panel, the International Panel on Biodiversity and Ecosystem Services, run by the UN, basically treats biodiversity and ecosystem services as kind of the same issue. And then the largest conservation organization in the world, the Nature Conservancy, really argues that the reason we need to conserve biodiversity, or a major reason, is because of all the ecosystem services that biodiversity provides. And this is just the former CEO of TNC and his book Nature's Fortune, which makes that argument. So the ecosystem service I work on is pollination by wild bee species. There are about 500 bee species native to the region, my study region, which is sort of northeast US. And by the way, the honeybee is not a native species. It's a exotic, invasive, domesticated species. And pollination is an important ecosystem service because about 85% of the world's plant species require animal-mediated pollination. And wild bees do roughly half of the crop pollination in North America also. Okay, so this is sort of a method section for the whole talk, because the data I'm going to show you were all collected in the same basic way. So to measure biodiversity and ecosystem services in the real world, we go out and net bees that are pollinating flowers of our study plant, whatever plant that is. And then we bring these bees back to the lab and identify them to the species level. So this is basically our measure of the biodiversity. It's the community that is doing the ecosystem service. And then in a separate set of experiments, we take unpollinated flowers and take them out to the field and let a bee of different types of bees visit the flower just once to pollinate it the way that it normally would. And then we take those flowers back to the lab and count the number of pollen grains deposited under a microscope. So that's our measure of sort of per capita function. And so then this is a really powerful method because we can actually measure the ecosystem function as it's delivered in the field by each species in the community. So we do that by just multiplying the number of individual bees we got of each species times the pollen grains deposited per individual. And that's just the pollination of the ecosystem service from that bee species. And then the sum across species is the total ecosystem service. So we've measured all of these data now for 6 different plant species, 3 wildflowers and 3 crops. So those are the data that I'll be showing you. And this is just sort of an overview of our study system. These are our research sites that we used for these data sets. And you can see they're kind of laid out between the cities of New York and Philadelphia. And these data sets I'll be showing you contain altogether about 7,500 individual bees of 110 species and over 700 of those pollen deposition experiments. So here I'm just showing you to kind of give you a sense of what the data look like, the data just on the biodiversity part, the bee community part. These are just 5 arbitrary sites from our watermelon study. And the pies are just showing you the bee community there. So the different colored slices are different bee species, and this is showing their relative abundance. And the total size of the pie here is aggregate abundance, so the total number of individual bees you had. So I just want to make a couple, like point out a few obvious things about these data that are really important to the BF relationship. One is that the size of these pies, the aggregate abundance, varies a lot across sites. So this site had way fewer bees than that site, for example. Another thing is that there tend to be big slices, a few big slices, these are the dominant species, and then lots of skinny little slices, these are the rare species. And then the third thing is that the identity of the dominant species, these bigger slices, tends to be similar across sites. This is blue-green species, for example, in these 5 sites in a lot of cases, but it's not always the same. There's some switch up in the identity of the dominant. Okay, so the question we asked of these data is what drives the difference in pollination between sites? And these sites were actual commercial watermelon farms. So this is it's a real ecosystem service. So we did this using a modification of the price equation from evolutionary biology. Modification was developed for BF by my collaborator, Jeremy Fox. And basically it does pairwise comparisons. So it starts with the difference in function between the 2 sites. So say this farm got more pollination than that farm. The price equation just starts with that difference in function. And then it partitions that difference into components that are attributable either to changes between the bee communities, these are the bee communities now, changes in terms of species level stuff, differences in species between the 2 sites, versus just differences in abundance of species that are present at both sites. In other words, shared species. So the idea here is the differences in species could be either species richness, this site just has fewer species than that site, or composition or composition. Obviously, these all go on at the same time. This site just lacks some of the colors that are at this site. And then the differences in abundance are differences that could occur even if the 2 sites, the differences that occur even for the species that are identical to 2 sites. So here I'm just showing you the case where the whole community is absolutely identical. This one just has fewer individuals. So it's based on shared species. Okay, so what we found in this paper is that in terms of explaining the difference in ecosystem service between farms, the effect size of abundance is 6 times as big as the effect size of all those species level things put together. So it's really driven by abundance. So I want to take a few minutes here to talk about why, about what I think the mechanism is, because I actually think it's pretty profound and could have ramifications for other types of ecosystem functions. So here I'm showing you what I call species function distributions for these 3 crops. So that's watermelon, cranberry, and blueberry. And the pollination ecologists, we only need the flowers, but I gave you guys the fruits. So basically what these figures show is they're just frequency histograms combining all the data for each plant and the whole bee community for each plant. They show the function provided here. This is the total pollen grains deposited per bee species. And this is just the number of bee species that deposited that amount of pollen. So I call these species function distributions. And they're very similar to this underlying species abundance distributions for these data anyway. And I'm sure you guys are all familiar with the species abundance distributions generally do look like this. They're these super skewed things with a few common species and many rare ones. And the correlation in these data sets is on the order of Pearson's R, it's like 0.8 usually. Okay, so I want to make a few observations about the shapes of these species function distributions. So the first is just that there are a few functionally dominant species that are doing most of the pollination. So they're circled in red here, and they're just depositing way more pollen than the other species. The second is that there's a very large number of species that are not doing much pollination. And these are, in fact, the rare species. They're often represented by one to a few individuals, and they just aren't doing that much functionally. And what we sort of the take home from this is a bit different for these first two crops as opposed to the third. So I'm just going to talk about these first two first. So here it's actually the same species here. That's Bombus impatiens, a common eastern bumblebee that is by far the dominant pollinator. And this bee is found at essentially all sites. It's both abundant and widespread. That's a strong correlation in ecology. So it doesn't account for any of the changes in species richness or composition between sites. It can't because it's always there. But it fluctuates in abundance. A number of individuals varies up to 100-fold across sites. And this species, this common species, therefore drives the changes in pollination across sites. Remember, this is a pairwise thing that's looking at changes between sites. So conversely, these rare species, being rare, they are not found at many sites. They're often like one or two individuals in the whole data set, actually. But the number of rare species a site has is what drives the species richness at that site, because these are kind of swapping in and out across sites. So rare species drive the changes in species richness. So I really think this is sort of a profound point here, that ecosystem services are decoupled from biodiversity simply by the shape of this species function distribution, because the common species are driving the function, the rare species are driving the richness, and therefore biodiversity is not driving ecosystem services. And again, I think this could have wide generality if, in general, species function follows from their abundance. Again, it does in my data. It doesn't mean it does in all data, because the species abundance distributions is super well known and has been for an extremely long time. They look like this. They're extremely skewed. Okay, so I said that this third plant was a bit different, so I'm now going to explain how it was different. So for blueberry, there are more of these dominant species. There are about 9 of them that were important pollinators. And so in these data, species richness does predict function, but it predicts it only like how many of these 9 species a farm has, predicts its pollination. And so that's still 9 out of 40 or so species. All right, so that was sort of an intuitive explanation that I came to based on looking at the distribution of the data. I now want to give you a more rigorous analysis. It was done by Mark Gannon, who's a former postdoc with me. He's now heading his own lab at University of Louisiana. So what Mark did was he got the data from some of the leading biodiversity ecosystem function experiments in the world, and then also collated all the data that's available globally that has basically has the parameters that we need in order to use the price equation. And these are biodiversity ecosystem service data on a variety of services. So kelp biomass production, natural grassland biomass production, pollination data sets come from my lab, and then carbon storage in tropical forests. So what I'm showing you here, both of these have the same axes. So on the X is functional dominance going from low to high. So low functional dominance just means that many species contribute to the function. It's sort of more even distribution of functional contribution. High functional dominance would be like what we were looking at. That they're just one to a few species that really do most of it. And then the y-axis is the effect of species loss on function. So that's basically how strong is the biodiversity ecosystem services relationship. So if there's a big effect of species loss on function, that means you lose species and you lose a lot of function. So that's basically strong biodiversity ecosystem function relationship. If you're down here, it means that you can lose species and it doesn't really matter for function. So here is what we found. And so I want to make two take-home points from this about the difference between these two plots. So for the first thing is the real world data sets have a wider range of the extent of functional dominance. And in particular, there are quite a few real world data sets that have high levels of functional dominance that don't show up at all in the experimental data. And then the second point is that this is a negative, we get a negative slope here for the real world, which means that you can predict how important biodiversity is to function by the level of functional dominance in the community. So when you have low functional dominance, in other words, more even distribution of function per species, when you lose species, you lose a lot of function. It makes sense. When you have high functional dominance, you lose species and you don't lose much function. And that's because you are more likely, again, this is not something an ecologist would argue with. In the real world, you're more likely to lose rare species than common species, right? When biodiversity declines, it's often the rare species that disappear. And if they're not functionally important because there's high functional dominance, then you lose biodiversity. It doesn't really matter. In contrast, in the experiments, that is not the case. And the reason, again, I think is due to experimental design largely. These are randomly assembled, researcher-assembled communities that tend to have lower dominance, but also importantly, there's no opportunity to quote unquote lose the rare species first, even though species loss, global biodiversity loss, is the intended scope of inference for the whole BF field, I think anyway. You can't really lose rare species first if they're just being put in there randomly by the experimenter, right? Whereas in the real world, you do. Okay, so the take home here is that in nature, the extent of dominance predicts whether biodiversity is important to ecosystem services or not, whereas that hasn't really been captured very well by experiments and theory. All right, so before leaving the topic of dominance, I want to mention something I've just been working on recently that I actually think is really cool, that has to do with the Pareto principle. So this is the Pareto principle. Basically, it's a popularized concept that just says that 80% of the consequences often come from 20% of the causes. And it holds true across a really wide range of things, like 80% of infectious disease transmission comes from 20% of infected individuals, 80% of earthquake damage is caused by 20% of earthquakes, 80% of NBA points are made by 20% of players, et cetera. And this is just a schematic I stole from a business management website. But basically, what I was wondering about this, well, this is probably obvious to all of you guys, but the Pareto principle just results from extremely skewed frequency distributions, right? I mean, it comes from, well, Pareto is one, but it's just one form of power distribution. It generally comes from power distributions, or log normal is the other common one. And in fact, power and log normal are what fit those species function distributions I was showing you. So I wondered whether, in fact, biodiversity and ecosystem function might follow the Pareto principle. So if that were true, according to the schematic, it would mean that 20% of bee species, the inputs, do 80% of pollination, which is the output. And according to the Pareto way of thinking, that would mean that only 20% of species are important, right? So I went to the data to check that. This is for the first plant species here. This is proportion of bee species included. And these are I'm just adding up bee species here. This is a proportion of pollen deposition accounted for by those same included bee species. And I'm obviously I added them up in order of the most functionally important first. So this starts off as a steep accumulation here, and then it flattens out up there where I'm adding the rare species that weren't doing much. So I just drew in these Pareto lines for you here. So we found that 20% of the bee species do 87% of the pollination for that plant. So close enough. For this plant, Basilia tenacetifolia, we found that 20% of the bee species do 98% of the pollination. For this plant, Polemonium reptans, we found that 20% of the bee species do 79% of the pollination. So then I wanted to go to some ecosystem service that wasn't pollination, just to see if this might have a wider generality. So I used the tropical tree carbon storage data from Barrow, Colorado Island's 50-hectare plot, Panama. And I found that 20% of the tree species do 83% of the carbon storage. Now, to be honest, this really surprised me for carbon storage. And that's simply because for trees and carbon storage, trees are very different from bees, largely a plant-animal thing. They're indeterminate growers. They have a way more dramatic stage or size structure to their populations. And the species abundance distribution for these trees is not log normal. It does not map onto the species function distribution. So the fact that I still found this is not due to the species abundance distribution. And again, that's just for the obvious biological reason that a very large number of little sapling trees, which exist in that age structure, don't store much carbon, whereas one canopy emergent like literally stores many tons of carbon. So it's a bit of a mystery to me why this follows the Pareto principle. It's not the species abundance distribution. Okay, so before leaving this topic, just want to make the point that this approach I've been showing you as the Pareto principle, it's actually a broader way of thinking about things. It's used in many fields. In economics, these are called Lorenz curves, and economists traditionally add them up the opposite way. So this would be percent species included, starting with the rare species and then putting the functional dominance here, and percent function delivered on the Y. That's what it would look like. But I put in the 80-20 thing in red just to make the obvious point. It's the same curve, it just flipped over the one-to-one line, right? But these, what they call Lorenz curves in economics that are used to study inequality, are also used to create the Gini coefficient, which many of us probably use. It's basically an index of inequality, and that's just calculated as the area of A over the area of A plus B. So this is sort of, there's like a broader conceptual background. All right, so why does this Pareto principle stuff matter at all? So I think it matters because it's inconsistent with the way that biodiversity and ecosystem services are sort of viewed or marketed. So biodiversity is largely this 80% of rare species that aren't doing a whole lot. They're only doing 20%. And in other words, biodiversity, by this way of thinking, would be not important. That's the blue. Okay, so to sum up what I've shown you so far, we found that when we combine the data, we either look at one place in time or we combine the data across, aggregated across the data set. We find very strong dominance in function by just a few species. So in other words, biodiversity is not important to ecosystem function. And that is not a feature of scale. That's true at both small and large scales. It's a feature of aggregating the data into one big distribution. So in other words, it's sort of a feature of looking at the mean or the sum of function per bee species. So I want to spend the rest of the time talking about what happens when we then consider the variability, the variance in function per bee species across space and time. So this is getting into the idea of the insurance hypothesis, which of course underlies a lot of what we're talking about today, but it also underlies a sort of scaling up in space and time issue, I think. So the idea here is that rather than just combine all these data for this one plant's pollination, we're now going to say, okay, so at each of these 3 sites where the plant is, it needs pollination, and the bee community pollinating at these 3 sites is somewhat different. So we really can't just combine all these data and look at which bees are most important overall. We need to say, you know, these are whoever pollinates it here are important, plus whoever pollinates it here, plus whoever pollinates it here. So it's more of a minimum set analysis kind of thing. And that's how we address the issue of scaling up in space and time, which is what I'm going to be talking about for the rest of the talk. So this again is just showing you our study sites. These are for the 3 crop plants. And what we did in this analysis is we asked not just what bees are most important in some, but what bee species do we need to pollinate this farm, and then this farm plus this farm, and this farm plus this farm, and adding farms. So it's a minimum set analysis of what bee species are important. And what we found is that as you need to pollinate more farms, and this is going up to all 48 farms of all 3 crop species on the X, you need a larger number of bee species to pollinate each of those farms. And what the curves here, there are 3 different colors. These are, in order to do this analysis, we have to establish some threshold level of pollination that is needed at the farm, right? Because otherwise we can't do a minimum set analysis. And there's, I don't really have time to get into the threshold issue, but we used a range of thresholds of levels of pollination that might be important to a farmer. And so these are 3 of those thresholds. The purple one is obviously a higher one, and you need more bee species when you have a higher threshold that you need to achieve, which isn't surprising. And a really key thing, big difference between this result and the dominance results I've been talking about in the first part of the talk is that we now need many more bee species. So you can see for the purple threshold, we need about 80 bee species, which is most of the bee species we recorded in the entire study. That dashed line up there at the top is a total number of bee species observed at all of these farms, which is about 110. So when we consider this variation of the insurance hypothesis, we need a lot more species. So I want to show you now, just show you the data to sort of explain like what's going on under the hood here is actually kind of subtle. And I think the best way to convey it is just to look at the data together. So here I'm showing you these stacked bar plots. This is for each farm in each year. I forgot to mention each of these farms are studied in 2 years. So the units here are site years. So in each, this is one farm in 1 year. And the number of pollen grains deposited by each bee species at that farm are shown in the different colors, with the larger, the dominant pollinators, the bigger bars at the bottom. And then all these little bars at the top were bees that weren't, species that weren't really contributing that much pollination. And you can see we have super high biodiversity here in the system. And then this black line is the threshold that we use in the analysis that we said, okay, that's enough pollination. And basically our species accumulation curves count all the species that are below this black line for any farm as needed. They're the ones that go into that accumulation curve. So this is how we actually generate those curves that I was just showing you. So there are 2 things I want you to notice just about these stacked bar plots. And by the way, I put the pies we saw before basically quite analogous, but those were for the bee community composition, and these were for the pollen deposition by each bee. They actually map onto each other really closely. So there are 2 things I want you to just sort of eyeball about this. One is that there tend to be these dominant bee species. This is Bombus impatiens here, that common eastern bumblebee. But then the identity of the dominant is different at some farms. This needed the red one, this needed the orange one, so it changes sometimes. And the second thing to notice is there's wild variation in the total amount of pollination that farms get. Some don't get nearly enough, some get way more than they need probably. And again, this is driven by wild variation in aggregate abundance. So I now want to zoom in to show you what's going on, zoom in on the farms that didn't get enough pollination, or they were at or near the threshold. So you notice that for these farms, we would include all these species in our accumulation curve because if they aren't even getting enough pollination, or if they just barely are, all those species are necessary. But that's going to include some species that actually in absolute terms are really not doing much. They're sometimes just depositing a few grains of pollen. And conversely, at the high end, for the farms that got a ton of pollination, we only need 1 species. And again, this is just to underscore the point I was making earlier, that in the real world, aggregate abundance has a really big effect on the answer you get for the biodiversity ecosystem function relationship. Okay, so that was about space. We can do a similar analysis, sort of scaling up in time. This is the work of Natalie Lemansky, who was a postdoc with me, is now an assistant professor at Ramapo College. So basically here we used some time series data we have for watermelon pollination. These are 23 farms. There are the data points, each of which was studied across 6 years. And this is the number of bee species that were needed to pollinate in 1 year, to pollinate all the farms in 2 years, 3 years, et cetera. And the bottom line is it roughly doubles the number of species you need when you consider a longer time period. And that's just due to fluctuations in bee populations over time, basically. And then for plants and pollinators, there's another really important axis to consider, which is plant species. And this is an important sort of axis of beta diversity for pollinators, because pollinators tend to have certain plants that they like to go to. You know, the community of pollinators is somewhat different on different plants. So this is the work of Dylan Simpson, a PhD student with me. And here we looked at, we're just adding up number of plant species now on the X instead of space or time. And these were 17 plant species that we grew in monoculture plots in a common garden. So they're like exposed to the same bee community, but then the bees can sort themselves onto these 17 plants. And this is the number of bee species that you need to pollinate 1 plant, 2 plant, et cetera, up to all 17 plants. And you can see that also increases quite a lot, and increases about twice as much as you would expect by chance. And the by chance part is a null model with the error bars. Actually, all these analyses had null models. I didn't really have time to get into that, but there's obviously a chance component in here to do with sampling and whatnot. Okay, so that was the scaling up question. And I want to finish by talking a bit about our lab's work on response diversity. So here the question is just, can biodiversity stabilize ecosystem services against environmental change? And I actually think the underlying idea is kind of the same as the scaling up idea insofar as its insurance hypothesis and beta diversity and species niches. So I think we all agree, I hope we all agree at this point, that response diversity is diversity of responses to environmental variation among species contributing to the same function. So here my function is pollination of a plant species. And the environmental factors that we look at most often in my lab are just the 2 leading causes of biodiversity loss, which are land use change and climate change. So this is a paper by Dan Carabo, who's a postdoc with me. He's now an associate professor at University of Minnesota. And in this paper, we looked at the bees pollinating cranberry farms across a land use gradient. So it went from cranberry farms like this one that are surrounded mainly by natural habitat to cranberry farms that are surrounded by more cranberry agriculture, basically. And we found 55 native bee species. They did about 1/4 of the commercial cranberry pollination. The rest was done by honeybees. And what I'm going to show you here, these figures are showing percent agricultural land cover on the X. So we're going from natural to more intensively agricultural. And then on the Y is going to be the ecosystem service, just the number of pollen grains deposited by different bee species. And I'm going to show you data for the most important 5 wild bee pollinators. And you can see that some pollinators don't do well in agriculture, like the black one here, Bombus bimaculatus, declines as it gets more agriculture, that deposits less pollen. But some are fine with agriculture, like the green here, again, that common eastern bumblebee actually prefers more agricultural areas. And so the signature of response diversity here is that the interaction term between bee species and land cover, where the outcome is the pollination provided, that interaction term was retained in AIC model selection. So there is response diversity. We've also looked at climate change. This is work of Romina Rader, who again was a postdoc with me. She now heads her own lab at University of New England in Australia. And what Romina did was use some of our data on crop pollination to look at how different groups of bees are responding to just the natural temperature variation in terms of how much pollination they're providing under different temperatures. So here we're going to be looking at on the X, the time of day, because these crop flowers are open from sort of 6 in the morning till 2 p.m. So it's across that time of day. And then the temperature at the time of day when the data were collected. And this is for 8 groups of bee species, including the honeybee. And then the colored scale bars here are about how many bees of each type we saw visiting flowers under those conditions. So that's basically how good was that pollinator under that temperature condition. And the red is we saw lots of bees visiting flowers, and the yellow and white is they weren't really doing much. So here are the data for 8 groups of bees. And you can just eyeball this. There is response diversity, right? I mean, these pictures are different. So some bees do more of their pollination at warmer temperatures and some do more at cooler temperatures. And we found that when we sort of projected IPCC scenarios forward to hotter conditions for this region, the total pollination did decrease, but only by about 5%, even though some bees are predicted to go way down, others go up. So there was sort of a buffering effect. Now, as we've been discussing this whole week, the fact that we found response diversity doesn't necessarily mean that there's stabilization or that it's related to species richness. Those are different questions, but in any event, we found it. Okay, so I just want to sum up then what we've found in my lab, what the data tell us about this relationship between biodiversity and ecosystem services in the real world. So again, in experiments, species richness, good predictor of function. In real world data, at least when you're looking at one place in time or when data are aggregated across places and times, we find that there's very strong functional dominance. And in fact, 20% or fewer of the species do 80% of the pollination. So there is not a strong biodiversity function relationship. However, when we disaggregate the data in sort of an insurance hypothesis type of way and look at different places and times or different plants, we find these increasing curves that you need more species as you scale up in space and time. And I think for analogous or maybe even the same reasons, we also find response diversity. And interestingly, there are interesting parallels to obviously the smaller scale version, but also to the larger scale version, just in coexistence theory. This is a little bit more sort of Peter Chesson storage hypothesis type of idea. So that is all I have to say. I want to thank funders, which has primarily been NSF, and thank you very much for everyone who's been involved in hosting the workshop, and my lab group, who they actually mostly work on stuff different from what I do, so I didn't have a chance to talk about it, but they're amazing, inspiring junior colleagues for me. And thank you so much for showing up.
Thanks, Rachel. That was a really, really interesting talk, and I think a very nice end actually to a series of talks over the month.
Great.
So we've got a couple of minutes for questions. I'm going to start with Laura. So Laura.
Thanks for a wonderful talk, Rachel. I loved it.
Thank you. Thank you.
We're finding is this on? Yeah. We're finding really similar things in plant communities as well.
Super
so lots
cool.
to talk about. So I wanted to ask a question to tie what you just showed with your idea for thinking about impacts of heat waves and extremes. So do you have a sense of if the dominant or rare species are going to be more or less vulnerable to the extremes, and how that
I have no idea. Yeah, I really don't know yet. Yeah, I mean, thank you for bringing up this heat wave thing, because that's like the grant I'm writing right now. It's totally a big front and center for me. Yeah, we really have no idea, and I don't think there'll be a general answer to that, because the identity of the functional dominant varies depending on where you are and what plant you're looking at. Like, I don't see any reason why the functional dominant in a very general way should, be tolerant of heat or not. Like, different families of bees are more or less tolerant of heat. Yeah.
Thank you so much, Rachel. It was a really good talk, very inspiring.
Thank you.
I was thinking about this 20-80%, I forgot the name now. So what about the supporting species? I guess that's another way of thinking about it. If you go outside this specific function, there must be a lot of supporting species and their biodiversity.
You mean not looking at what's pollinating this plant?
Yeah, I'm thinking like your example with a soccer player or what it was. The other some soccer players have to be goalkeepers or something like that.
Yeah. Well, Right. So bees, I could say a lot about how pollination as an ecosystem function is different from what people often are thinking for functions, partly because I think the whole field is conditioned on a small number of study systems, namely plants and microcosms, microbial things sometimes. Bees don't necessarily support each other in pollination in the same sense that basketball players would. Bees are, here's a really interesting thing. Pollination from a bee's point of view is a mistake. So bees are trying to collect pollen. They eat pollen, it's their protein source. So bees, they're kind of herbivore wasps. Wasps do these horrible things of feeding their offspring paralyzed caterpillars and all that kind of stuff. And bees don't go there. They only collect pollen, which is their protein source. And long way of saying they're not looking to pollinate anything. Like the pollen that pollinates, that gets deposited on another planet, is pollen that they failed to like stick on the right part of their body and take back to their nests for their larvae. So they don't really support each other in pollination.
I was thinking more in the wider sense of this with biodiversity, how it's communicated, that maybe, I mean, there are plants supported the pollinators and
sure, absolutely.
Some plants need to be supported by other plants, etc. So
absolutely. No, in the larger sense.
This,
no, absolutely. And in talking to a public audience, that's insofar as you kind of have to have like one message. I believe that would be the right message. But to a scientific audience, I'm addressing like what ecology says, and I think we have a ways to go.
Okay, thank you for a wonderful talk, really very inspiring.
Thank you.
So you've shown some time series of the number of species through time. Mm-hmm. Have you tried to understand where these trends come from? And for example, if there's even at the population level, whether there's some like predictable pattern, maybe based on their pollen usage?
Right. I mean, so that comes from, the variation over time comes from qualitatively from two places in bees. One is just the normal, like it's a biological thing, just the phenological turnover over the course of the season. If a plant blooms for a longish time, like many crops do, because that's an artificial thing. Like in nature, plants typically would bloom for like 3 weeks-ish, but farmers stagger their plantings because they want to be bringing watermelons to market all year. So many things I've learned in doing this research, which is great experimentally because we have this experimental system set up for a longer time period. So there's a natural part of that variation comes from natural turnover in the bee species, because the bees will be active for maybe like 6 weeks each, and then you get another species coming in. So some of the temporal turnover is biological turnover in the part of the bees, but across years, which was the plot I showed you here, we don't know where that is. We assume it's stochastic. And I mean, there's like weather. I mean, these are insects, right? But there's huge variation in insect communities year to year.
Yeah. Thanks.
Maybe shall we ask over the break? Is that right, Ceres? So yeah, please join me in thanking Rachel once again for a fantastic.
Thank you.
For those of you in the workshop, let's meet back about 5 past, 10 past-ish.
20%.
Well, they would, because there's a much more complicated. Oops, I'm going to do a smart thing and turn off my mic.